Welcome back to Biology 2225 at CNM. We are discussing Chapter 23, the urinary system. Last time we left off at objective number 14, and so we've already described the location of the kidneys, their approximate shape and size, and we've talked about the various protective coverings in which the kidneys reside. The renal fascia just beneath the parietal peritoneum, the perirenal fat capsule that cushions the kidneys and generally holds them in place, and then the collagenous fibrous capsule that covers the surface of the kidneys themselves, and the collagen fibers that extend from the capsule to the fascia that again help hold the kidneys in position in a retroperitoneal position in the abdominal cavity. Here we see another picture again of the kidneys in their retroperitoneal position and the abundance of perirenal fat tissue, adipose tissue, that again essentially just fills space, holds them in place, and cushions and protects the kidneys. Here we see the shape of a kidney again with a convex surface, lateral surface, and a concave medial surface, and then the slit-like hilum through which lymphatics, blood vessels, nerves, and the ureter communicate with the kidney. Now, when we talk about the internal structure of the kidney, in other words, what's beneath the capsule, the fibrous capsule, we have what's called parenchyma tissue. Parenchyma tissue, or the renal parenchyma, are the actual functional cells of the kidney forming this C-shaped region right here. This is known as the renal parenchyma. And then deep to the renal parenchyma, you can see that you have a series of hollow spaces, and this is all referred to as the sinus of the kidney. This more hollow area here underneath the parenchyma is referred to as the sinus. And the sinus consists of blood vessels, some of which you're seeing here, lymphatic vessels, nerves that will help regulate urine production, and a variety of urine collecting structures, which are these larger hollow tubes here. Most of the space between these structures I've just mentioned is filled with adipose tissue. All right, so as we just said, we've got the renal parenchyma, which again is the actual cellular tissue of the kidney itself that forms urine, and then the deeper sinus containing all those structures that I just mentioned. Now, the renal parenchyma, the functional cells of the kidney, is divided up into two layers, and this is a typical arrangement for most organs. The outer layer is referred to as the cortex, and the deeper layer underneath the cortex is referred to as the medulla. Both layers are involved in urine production, but the majority of the kidney's physiology occurs in the cortex layer. The cortex does have extensions that dive down into the medulla. These extensions of the cortex are referred to as renal columns, and they extend down medially towards the sinus. In between the renal columns is the actual tissue of the medulla itself, and it is arranged into approximately 6 to 10 cone-shaped structures known as renal pyramids. In 2 dimensions, they look like triangles, but in 3 dimensions, they're more cone-shaped or pyramid-shaped. And so renal pyramids are the actual functional tissue in terms of urine production. That's the functional part of the medulla. They taper to a point at the sinus end, and you have the tips of these renal pyramids are referred to as the renal papillae. A typical lobe, what we call the lobe of a kidney, consists of one pyramid and the cortex that sits on top of it. That is referred to as the lobe of a kidney. As we said, the sinus, it consists of a variety of vessels and hollow spaces. At the tip of each pyramid, in other words, at the papilla of each pyramid, there is a hollow space that is designed to collect urine that drips out of the papilla once it's formed. And that little cup-like hollow space that nestles the papilla of each pyramid is referred to as a minor calyx. So once urine collects in the minor calyx, the physiology of urine production is complete. That happens up in the cortex and the medullary pyramid. Once it starts to accumulate in the minor calyx, now really the task is just to transport that urine to the storage site, to the urinary bladder, until it's time to release it from the body. So once we get to the minor calyx, in other words, there's not much physiology left other than transport to the bladder. Minor calyces are found at the tip or papilla of each pyramid. Minor calyces will then fuse together to form major calyces. That's calyces, by the way, is the plural of calyx. So two or more minor calyces will fuse together to make a major calyx. And then all the major calyces of the sinus will fuse together to create one common collection site referred to as the renal pelvis, which is centrally located in the sinus near the hilum. The renal pelvis is a funnel-shaped hollow space that eventually funnels down and leads to a single tube which exits the kidney, and of course we call that tube the ureter. The ureter is, as you see here, just really a continuation of the pelvis, but it is the tube that then drains the urine away from the kidney and ultimately to the urinary bladder. Here we see a diagram of that structure we just talked about. Again, the cortex, where most of the physiology occurs, forming the outer layer of the parenchyma, the functional tissue, and the medulla organized into these triangular structures known as the renal pyramids. In between the pyramids are extensions of the cortex known as the renal columns. And here we see it diagrammatically. Again, the cortex on the outside and the columns, renal columns, being between the triangular renal pyramids. Again, remember, in 3 dimensions they're actually cone-shaped. The tip of each pyramid is referred to as the renal papilla. This is where urine will leak out once it's formed, and this hollow space is the minor calyx. You see that 2 or more minor calyces will fuse together to make a major calyx, and 2 or more major calyces will eventually fuse together to form a common renal pelvis. As I mentioned, the renal pelvis is funnel-shaped. All the urine passes through the pelvis and eventually into the tube that we call the ureter, once again to carry the urine to the bladder. Here again we see all the various structures we've just talked about highlighted, and we've pretty much talked about all of these structures. Another diagram of the kidney, labeled the diagram of the kidney, once again showing all the structures that we've just discussed. Alright, so under normal conditions, the kidneys receive about 1/5 of cardiac output. That's quite a bit of blood, considering that the kidneys only constitute less than 1% of the body's mass. So why do the kidneys get so much of the body's blood? Well, it has to do with the kidney's function, of course. Again, the function is to filter blood and maintain blood chemistry. And so the kidneys need to receive a good portion of the body's blood volume to serve this function, to carry out this function on a continuous basis. As we know, flow to the kidneys can increase or decrease depending on the body's needs for blood elsewhere, such as during exercise. But under normal resting conditions, we typically devote about 1 1/5 of our cardiac output to the kidney so that they can perform that filtration function, that processing function on an adequate amount of blood. So the blood supply to the kidneys is an important element of kidney structure that we need to understand. The supply of blood to the kidney is a branch of the aorta. Remember, the aorta is the main systemic artery that comes out of the heart. We pass through the aortic arch and then to a descending aorta. The descending aorta, as the name implies, descends or travels down the thoracic cavity, passes through the diaphragm, and ends up as the abdominal aorta once it gets into the abdominal cavity. This abdominal aorta gives rise to many various arteries that serve the abdominal viscera. The artery that comes off the abdominal aorta and sends blood to the kidneys is referred to commonsensibly as the renal artery. And then it's just a matter of the renal artery dividing up progressively into smaller and smaller vessels, serving more and more localized regions of the kidney as it divides. So a renal artery will divide into a few segmental arteries, and then each segmental artery will divide into a few interlobar arteries. Interlobar arteries are always located in the renal columns between the medullary pyramids. And remember, a lobe of the kidney is a pyramid, and the cortex that sits outside of it, external to it. And so an interlobar artery is a good name because it's between the lobes of the kidneys, in other words, in the renal columns. Interlobar arteries will carry blood out towards the cortex of the kidney. Once the interlobar artery arrives at the boundary between the cortex and the medulla, it will curve around, essentially do a 90-degree gradual curve, and become an arcuate artery. The arcuate artery sits in the boundary between cortex and medulla, and it has generally a curved shape to it as it goes from column and again goes through this 90-degree curve to its new orientation. And so arcuate refers to that arcing pattern or arcing shape of the artery. As the arcuate artery travels along the boundary between cortex and medulla, it will radiate arteries, branches of itself, up into the cortex. And these are known very logically as cortical radiate arteries. Again, branches of the arcuate artery that radiate outward or upward to the cortex. So these are at this point fairly tiny arteries that are investing or perfusing the cortex. Each cortical radiate artery will branch into many what we call afferent arterioles. So obviously the radiate arteries are tiny because their branches are arterial. So by this time we're talking about very small blood vessels. And so we have many afferent arterioles branching off of the radiate arteries. Remember, afferent, when we talked about the nervous system, afferent means passing into something. And so these afferent arteries are going to carry blood into something important. What they are supplying blood to is a structure known as the nephron. The nephron, as we will see, is the functional unit of kidney structure. This is the microscopic functional unit of the kidneys where urine is actually produced. And it all begins, of course, with filtering the blood of impurities so that it can be processed. So these afferent arterioles are going to supply, as arterioles always do, they are going to supply a capillary bed where this physiology of filtration and reabsorption begins. That initial capillary bed that is involved in the first process of urine production is referred to as a glomerulus. And so each afferent arteriole will supply blood to a capillary bed known as a glomerulus. Glomeruli, which is plural, are interesting capillaries. They're actually spherical, little ball-shaped capillary beds. And in fact, the word glomerulus means little ball of yarn. which is what the early anatomists thought it looked like when they glimpsed it under a microscope. Now, the glomerulus is only the first capillary bed in the renal circulation. There is going to be a second capillary bed as well. So once again, we see that we are dealing with a portal circuit. Remember, a portal circulation is one in which there are two capillary beds in sequence instead of just a single capillary bed. And we've hinted at this before, and we've referred to it earlier in the semester when we were talking about blood vessels. And at the time, we mentioned that there were three portal systems in the body. The biggest one is the hepatic portal system. Remember, where blood travels from many of your visceral organs to the liver for processing. There's a second portal system between the hypothalamus and the pituitary gland, which we talk about when we talk about the endocrine system. And this is the third portal system of the body, the renal portal circulation. And so blood is, and basically the rationale for why we have a portal system here is that different physiological processes will be occurring at these two capillary beds. So at this first capillary bed, the glomerulus, the main function, the main physiological process that we're interested in occurring here is something called filtration. And you may recall, again, back in our initial discussion of blood vessels, that filtration is the movement of materials out of a capillary into the tissue space. And we said that when blood flows through capillary beds, generally speaking, you get filtration at the front end and reabsorption at the back end. And that was due to changes in blood pressure as blood flows through the capillary bed. And so filtration was movement of materials out through a combination of physical hydrostatic force and osmotic forces. Remember, reabsorption, which tends to occur at the back end of a capillary, reabsorption is the movement of materials in the opposite direction from the tissue space back into the bloodstream. And again, it's driven primarily by hydrostatic forces and osmotic pressures. In the glomerulus, we only get filtration. We get almost no reabsorption. And the reason for that is, as we'll see in a few slides, the pressure in the glomerulus is higher than it is the pressure in most other capillary beds in the body. So we want the pressure in this particular capillary bed to be high, because what we want to happen at this glomerulus is filtration through its entire length. So even though it might lose some pressure as blood flows through the glomerulus, by the time blood gets to the end of the glomerulus, the pressure is still high enough to cause filtration rather than absorption at that back end. And so this high pressure causes more filtration to happen at the glomerulus than at any other capillary bed in the body. And so this is the primary function that takes place, physiological process that takes place at the glomerulus, is filtration, movement of materials from the blood into some tissue space. And again, this is the beginning of urine production, because in order to regulate blood chemistry, one of the things we have to do, of course, is to remove unwanted materials from the blood. We have to remove any materials that are in excess, glucose, sodium, etc. And this removal of materials from the blood in order to maintain proper blood chemistry is called filtration. And again, it happens at this first capillary bed, the glomerulus. Now, since this is a portal circuit, we have to have a vessel that carries blood from the glomerulus to the second capillary bed in the circuit. And in this particular case, the vessel that drains the glomerulus and leads to the second capillary bed is known as the efferent arteriole. And of course, efferent, remember, means outgoing. So afferent arteriole, incoming, efferent arteriole, outgoing. And of course, going into and coming out of the glomerulus. You'll notice something interesting here, pretty unique in the body, is that we have a capillary bed that is being drained by an arteriole. Everywhere else in the body that I'm aware of, capillary beds are drained by venules. But this capillary bed is drained by an arteriole. Even in the other portal systems, the first capillary bed is pretty much drained by a venule or a vein, not an arteriole or an artery. The functional significance of this is that, remember, arterioles have thicker tunica medias than do venules. Arterioles are more resilient in terms of the durability of their walls and our ability to alter their diameter. And so what this means is, through vasoconstriction and vasodilation, we can alter the size of these arterioles. If we vasoconstrict these arterioles, in other words, make them smaller, and by the way, the efferent arterioles are smaller than the afferent arterioles to begin with anyway, but we can make them even smaller through vasoconstriction. What that does is that imbalance between afferent and efferent arteriole means that more blood can enter the glomerulus than can exit it. What that does is it causes a buildup of back pressure. And so this is one of the primary reasons why the blood pressure in the glomerulus is higher than it is in other capillary beds. It's because it's the only capillary bed that's drained by an arteriole, which allows for the buildup of back pressure by the arteriole being smaller and through our ability to constrict it more than we could constrict a venule. And again, that's what we want. We want the higher pressure to promote filtration. All right, so where does the efferent arteriole carry blood to? Well, of course, it carries blood to the second capillary bed in the portal circuit. And for the most part, these are referred to as the peritubular capillaries. The peritubular capillaries. As we will learn, the nephron, the functional unit of the kidney, is really a long, convoluted, microscopic tube through which filtration and reabsorption are going to occur. And through most of its length, that nephron, the tube of the nephron, is surrounded by a capillary bed. And so peritubular capillary bed refers to the fact that this capillary bed is surrounding and it is in intimate contact with the tube that forms the nephron. Now, there is a part of the nephron that forms a loop that dives down into the medulla, because remember, at this point, we're up in the cortex. Remember, the afferent arteriole branched off the cortical radiate artery. So most of this vascularity that we've been talking about up here at the microscopic level is in the cortex. But there is a part of the nephron, a loop-like structure that dives down into the medulla. And so we also have a capillary bed that dives down into the medulla along with that loop-like structure. This capillary bed is referred to as the vasa recta, and it's a second capillary bed that is found in the medulla rather than up in the cortex, as would be the typical peritubular capillaries. All right, so once blood flows through the peritubular capillaries, we're going to have not filtration, that took place in the glomerulus. We're going to have the opposite physiological process known as reabsorption. So the glomerulus is pretty much devoted to filtration, movement of materials out. The peritubular capillaries are going to be primarily devoted to reabsorption, which is pulling materials back into the bloodstream. Why do we want to do this? Well, as it turns out, as we'll see, we filter way more materials out of the blood than we need to. In fact, we filter so much blood that if we didn't reclaim most of what we filter, we would die very quickly. So we have to reclaim much of what we filtered. Filtration is not a very specific process, so it's not just bad things that get filtered out, it's everything. Anything that can pass through the capillary wall and get pushed out by blood pressure gets pushed out, and that includes, again, many things that we don't want to lose in the urine. And so reabsorption at the peritubular capillary beds is equally important in terms of reclaiming vital plasma components And again, in maintaining proper blood chemistry by selectively reabsorbing, we keep the blood plasma at just the right chemistry. There is a third physiological process called secretion that takes place in the peritubular capillary beds. Secretion is like filtration in terms of the direction of movement, but it's much more selective. So whereas filtration is a very generic kind of movement and not very selective, secretion is movement of materials from the blood into the tubes of the nephron, but at a very selective basis, again giving us more control over the exact composition of blood plasma. But we'll come back to these three physiological processes later in the chapter and describe in more detail how they occur. So once the blood flows through the peritubular capillaries, we're pretty much done with the three major physiological processes of filtration, reabsorption, and secretion. And now it's time to get the processed blood back into the mainstream circulation. And so the peritubular capillaries are drained by venules into cortical radiate veins, which parallel the cortical radiate arteries. They run through the cortex and eventually drain into arcuate veins, which parallel the arcuate arteries and sit in that boundary between cortex and medulla. The arcuate veins then lead to interlobar veins, which are found in the renal columns between the pyramids. And the interlobar veins eventually fuse together and lead to a renal vein that drains the entire kidney. And of course from there the renal veins will dump blood into the inferior vena cava, which is in the abdominal cavity as well. The inferior vena cava carries blood upward, pokes through the diaphragm again, up into the thoracic cavity, where of course we then see it in a familiar position of draining blood back to the heart, specifically into the right atrium. And now we're back to the mainstream circulation. Here we see a diagram of the various vessels that we've talked about, highlighted on a cadaver kidney. So just to review, a renal artery supplies blood. It divides quickly into segmental arteries. The segmental arteries then divide into interlobar arteries, again specifically located in the renal columns between pyramids. The interlobar arteries will arc around and become arcuate arteries that sit in this boundary again between medulla or pyramid and cortex. Here we see a diagram of what we've just talked about, and again this is a more thorough picture of what's happening. So again, the renal artery, segmental arteries, interlobar arteries between the pyramids arcing around to become arcuate arteries. And here, if you look very closely, coming off the arcuate arteries, you can see the cortical radiate arteries, these little red branches radiating up into the cortex. Of course, at this point we're getting microscopic. That would lead to the glomerulus and the peritubular capillaries associated with the nephron. The peritubular capillaries would then, of course, drain into the cortical radiate veins, these tiny blue vessels also up in the cortex. The cortical radiate veins then become the arcuate veins, again sitting in the boundary between cortex and medulla. Arcuate veins drain blood into interlobar veins between the pyramids, and the interlobar veins will join together to form the renal vein, which passes into the inferior vena cava. The part we did not see was that microscopic circulation of the kidney on that previous diagram. So this shows us the microscopic elements of that circulation. Remember, we have the arcuate artery here. Just to orient you, this upper region here is the cortex, as you can see, and then down here we have the medulla layer of the parenchyma. So this represents the boundary between cortex and medulla, and as we said, the arcuate artery sits nicely in that boundary, arcing around that boundary between cortex and medulla. The arcuate artery sends the cortical radiate arteries up into the cortex. So these are two cortical radiate arteries, and each cortical radiate artery supplies branches several branches, what are known as afferent arterioles. And the afferent arterioles are going to feed the first capillary bed, this little spherical ball-like capillary called the glomerulus. And again, this is where filtration is going to take place. And you'll notice, as we'll talk about in just a little bit, the glomerulus is surrounded by a cup-like structure that's going to collect the fluid that gets filtered out of the glomerulus. And that leads to, as I mentioned earlier, a tubular structure known as the nephron. And so all this tube here, this long coiled tube, is the nephron, or at least part of the nephron. From the glomerulus, we have a vessel that drains blood out of the glomerulus. Remember, this is the unusual part. Instead of being a venule, this is an arteriole known as the efferent arteriole. The efferent arteriole then feeds a second capillary bed known as the peritubular capillary bed. And as I described, the peritubular capillary bed is intimately arranged around the tubes of the nephron. I did mention that there, in some cases, there is also, or there are branches of the afferent arteriole that will dive down into the medulla, surrounding the loop-like portion of the nephron, and that set of capillaries that surround the loop-like portion will be referred to as the vasa recta. The vasa recta. Once blood is done flowing through the peritubular capillary bed and all the processing is complete, the venules will drain the peritubular capillary bed, and those venules will lead into the cortical radiate veins, as we saw in the previous diagram. And the cortical radiate veins will then drain blood into the arcuate vein, which sits again parallel to the arcuate artery. And of course, they will lead to the interlobar veins and ultimately onto the renal vein. So we've talked about the overall gross anatomy of the kidney, and we've described the blood circulation of the kidney. And it's now time to talk about the microscopic anatomy of the kidney, and specifically the functional unit of the kidney that is known as the nephron. The nephron. This is where all the physiology takes place and ultimately where urine is produced. Each kidney has about 1.2 million nephrons, so obviously they are microscopic in size and abundant. Each nephron consists of two major parts, the renal corpuscle and the renal tubules. The renal corpuscle is that first part of the nephron that involves the glomerulus, and this is of course where filtration takes place, as I mentioned. The renal tubule is the rest of the nephron, the long coiled tube that we saw, that essentially processes that filtrate that's produced at the renal corpuscle and turns it into urine. Again, this processing that turns the filtrate into urine is done by, as I mentioned, reabsorption and secretion, which we again will take a look at later in the chapter. So we're going to start by looking at the renal corpuscle, the first part of the nephron. And the renal corpuscle consists really of two parts: the glomerulus, the capillary bed that we've previously described, and a cup-like structure that I referred to that surrounds the glomerulus. This cup-like structure is referred to as the glomerular capsule. The glomerular capsule. It's a cup-like structure that surrounds the glomerulus. Now, the glomerular capsule is not a single layer of cells. In fact, what it is it's a double layer of cells. It's a double layered capsule with a hollow space in between the two layers. The outer layer of the capsule, which is called the parietal layer of the glomerular capsule, is just simple squamous epithelium. So this is the outside of the capsule. This part is not in direct contact with the glomerulus. It forms the outside of the glomerular capsule. The part of the glomerular capsule that is in contact, or at least close to the glomerulus, the capillary bed, is the inner lining or inner layer of the glomerular capsule, known as the visceral layer. The visceral layer. And this inner layer that's close to the glomerulus consists of very specialized cells called podocytes. Podocytes. And what's interesting about podocytes is that they don't form a typical single layer of cells like a typical simple epithelium would be. Instead, the podocytes are form-fitting to the capillaries, form-fitting to the glomerulus. So in other words, the inner layer, this visceral layer of the glomerular capsule, is not a single sheet-like membrane of epithelium. Instead, it's a highly irregular, complex layer of cells that completely surround every capillary of every individual tube capillary of the glomerulus. So you can't really give it a shape. Its shape is essentially the shape of the glomerulus. So the entire glomerulus is completely covered with the podocytes of this visceral layer of the glomerular capsule. In between the two layers, I said there was a hollow space, and this is referred to as the capsular space. And generally what happens here is that the fluid, the material that's filtered out of the blood, called the filtrate, accumulates in this space between the parietal layer and the visceral layer. So here we see a visual of what we've been talking about. Again, the larger afferent arteriole feeding blood into the glomerulus and the smaller efferent arteriole draining blood out. Again, that disparity helps create high blood pressure, and that high blood pressure tends to push materials out of the glomerulus, again called filtration. The cup-like structure that surrounds the glomerulus is the glomerular capsule. Again, the parietal layer of the capsule is the layer of simple squamous epithelium on the outside. And then the inner layer, again, very complex lining that completely covers the surface of every capillary in the glomerulus. And these are known as podocytes. And you'll notice the podocytes have highly irregular shapes, many arms branching out, and you'll notice the arms are somewhat serrated. And so the arms of neighboring podocytes with their serrated edges will interlock with one another. And so they form a very tight kind of covering to the glomerulus. And what this means is what's happening here is the podocytes are helping create a filtration membrane. So instead of just the simple squamous of the capillary doing the filtration, the podocytes also determine what can be filtered out by forming these so-called filtration slits between the arms of the podocytes. So in order for materials to pass or filter out of the blood, the material not only has to be able to pass through the squamous epithelium of the capillary, but it has to be able to fit or squeeze between the filtration slits of the created by these podocytes. So it gives us an extra level of regulation over filtration. As we said, once material does filter out of the capillary, out of the glomerulus and through the filtration slits, it then winds up in this hollow space between the parietal layer and the visceral layer, and of course this is known as the capsular space where filtrate accumulates. Notice, just as a preview, the filtrate doesn't remain here. Of course, that's a limited space, and filtration is occurring constantly. So as filtration occurs, pressure builds up, and that forces the filtrate to move into a tube that drains the glomerular capsule, and that tube is going to lead to the tubular portion of the nephron, where some more of the physiology takes place. So again, altogether, this is referred to as the renal corpuscle, and it consists of the glomerulus itself plus this renal capsule that surrounds, or glomerular capsule that surrounds the glomerulus. An older name, by the way, for this glomerular capsule is Bowman's capsule, and it's still very common to hear people refer to this capsule as Bowman's capsule. just to orient you, we're somewhere around objective 27, 28 right now. The renal corpuscle does have two opposite sides to it, as we saw. It's got a vascular pole and a urinary pole. The vascular pole of the renal corpuscle is the side where the afferent and efferent arterioles enter and exit. And then the urinary pole is the opposite side of the corpuscle, and of course that's where the, as I just mentioned, the tubular portion of the nephron begins as filtrate drains out of the capsule. Now, the renal tubule, actually I'm going to go back to the objectives here before we start talking about the renal tubule. Objective 30 asks us to describe the structure of the vascular pole and the urinary pole. And I just want to make sure that we understand that before we move on to the renal tubule. So again, the vascular pole is where the arteries come in and go out. And again, as we said, as we remind ourselves in Objective 30, the afferent arteriole is larger than the efferent arteriole, helping to create that high blood pressure. And again, the urinary pole consists of the tube that leads away from the glomerular capsule, carrying the filtrate away. I just want to mention that the urinary pole is formed primarily by the parietal layer of the glomerular capsule, which becomes the tube that then leads away. Once we get away from the glomerular capsule, that parietal wall converts from simple squamous epithelium into a simple cuboidal epithelium. And so the tube that leads away from the glomerular capsule, even though it's continuous with the parietal layer, is not the same kind of epithelium. It is composed of simple cuboidal by the time it becomes a tube. All right, so now we can talk about this renal tubule, of course, that originates at the urinary pole and begins the tubular portion of the nephron. It's the renal tubule, of course, is leading away from the capsule, and ultimately this tube, though it goes through many twists and turns and loops, eventually leads all the way to the papillae, or the tips of the medullary pyramids. And so remember, as we hinted at earlier, urine, fully processed urine, drips out of the tips of the medullary pyramids and is collected by the minor calyces. So somewhere between glomerular capsule and the medullary pyramids, as filtrate flows through this very long, relatively long tubule, it is going to be processed into urine, which then drips out into the minor calyces. So what are the parts of this tubule that are going to process what's been filtered and turn it into urine? Well, there are four components, four parts to the renal tubule, or the tubular portion of the nephron. And these four parts are the proximal convoluted tubule, the nephron loop, which honestly most people still refer to as the loop of Henle, and I believe I have given you that name in the objectives. Yes, in objective 32, you see it referred to also as the loop of Henle. And then after the loop of Henle, we have something called the distal convoluted tubule. So proximal and distal refer to how close we are to the renal corpuscle. The proximal convoluted is the closest part to the renal corpuscle. Distal convoluted tubule is a convoluted portion of the tubule that's farther away from the renal corpuscle. The loop of Henle is in between, of course. And these three parts are a part of a single nephron. And then there's a final part of the nephron called the collecting duct, and the collecting duct receives fluid from the distal convoluted tubule, but it doesn't receive fluid from just one nephron. The collecting duct is actually part of several nephrons, and so it's going to receive fluid from actually many distal convoluted tubules of many nephrons, and it will all join together, and it's the collecting duct that will then dive down into the medullary pyramid and make it all the way to the tip, where the urine will then leak out. So we say that the nephron is divided into 4 regions, but really a single nephron has only 3 parts to it, and then multiple nephrons combine to form that 4th part, which is the collecting duct. And I will show you a picture of this in a few slides. So we're going to talk about each part of the tubular portion of the nephron now in order. And we're going to begin with the proximal convoluted tubule. Again, this is the part that initially arises from the glomerular capsule. It does turn out to be the longest individual portion of the nephron, even though it doesn't look like it. Looks like the collecting duct would be, but because of its highly coiled structure, it's hard to gauge its length. So even though it doesn't look like the longest part necessarily, if you were to uncoil it and stretch it out, it would indeed be the longest. As I mentioned earlier, we get a transition from simple squamous in the parietal layer of the glomerular capsule to a simple cuboidal epithelium in the proximal convoluted tubule. One way to distinguish these cells from other parts of the tubular nephron is that the cuboidal cells of the proximal convoluted tubule have the most prominent microvilli associated with them. And so again, remember, microvilli increase surface area. They're tiny little projections, cellular projections. So we're used to hearing villi as structures associated with, say, the intestines. Well, villi are microscopic, finger-like projections in the wall or the lining of the small intestine. But a single villus in the small intestine consists of many, many, many cells. Microvilli are almost submicroscopic. They're actually difficult to see even with a conventional light microscope, but with an electron microscope we can clearly see what they are. On a light microscope they look like a fuzzy border to the cells, but under an electron microscope we can see that they are actually individual cytoplasmic extensions of individual cells. So if you had a powerful enough microscope, remember we're talking about cuboidal cells here, which are generally cube shaped with a nice round nucleus, but with the cuboidal cells of the proximal convoluted tubule, what we see is at the lumen, the luminal surface, and lumen is a term we generally use for the hollow interior of a tube-like organ. So the stomach has a lumen, the esophagus has a lumen, and of course the nephron tubes have a lumen, a hollow center portion. The cuboidal cell that lines the lumen will have all the typical structure of a cuboidal cell, but at its luminal surface it will have tiny little finger-like projections of its membrane, of its cell membrane. Again, these are too small to see clearly with a light microscope, but with an electron microscope we can clearly see what they are. They're not cilia, not to be confused with cilia. Remember, cilia are just protein structures, and cilia are capable of independent movement. These are not made of protein. These are actually just invaginations and evaginations of the cell membrane. There are tiny bits of cytoplasm inside them, and they don't have motor proteins in them for individual movement. And so these little folds are referred to as microvilli. And so as I hinted at earlier, the role of microvilli in general is to dramatically increase surface area. And so in this case, the increase in surface area is going to be very important so that we can do the amount of reabsorption that we need to do. Remember, filtration, movement out of the blood, takes place in the glomerulus. We have to recover the vast majority of what's filtered or we would die. And so we need great surface area to reabsorb most of what's been filtered out from the glomerulus. All right, the second part of the nephron, again, is the loop, what's called the nephron loop, or I'm just going to continue to refer to it as the loop of Henle. The loop of Henle is this U-shaped portion of the nephron that is the only part of the nephron that dives down into the medulla, other than the collecting duct. But if I'm looking at a single nephron, just the first three parts, the only part of an individual nephron that dips down into the medulla is the loop of Henle. It is broken up into a descending limb and an ascending limb. Of course, a descending limb is the part on the proximal convoluted tubule side that carries fluid filtrate down into the medulla. And then the ascending limb carries the fluid back up to the cortex. The descending limb and ascending limb have what are called thick and thin segments to them. The thick and thin segments are different in terms of their function and structure. The thick segments are constructed again from simple cuboidal epithelium, and the thick segments tend to form the beginning of the descending limb and almost all of the ascending limb. All right, so the very beginning of the descending limb and almost all of the ascending limb is what we call the thick segment. And the cells of the thick segment are, as you can see here, involved in active transport of salts. And again, we're not gonna go into too much detail about why right now. That comes later. But just know that transport of salts is going to have something to do with osmosis, creating environments that have high osmotic pressure and low osmotic pressure. And so that's going to have something to do with water, the movement of water. Because they do so much active transport, of course, you find many mitochondria, because active transport is an ATP requiring activity. So the thick segment is involved in active transport of salts like sodium and chloride, and that's going to affect osmosis. The thin segment is called a thin segment because it literally has a thinner wall made of simple squamous epithelium instead of simple cuboidal. You generally find the thin segment at the lower part of the descending limb and forming the actual loop, the U-turn portion of the loop of Henle. And this region, the thin segment, ends up being very permeable to water. So the cells are full of aquaporins, again, those passive channels that allow water to move very freely into and out of the cells. So we get active pumping in the thick segment, and we get just passive movement or osmosis of water in the thin segment. Again, we're going to have to wait a little bit to put this all together to see what the functional significance of all that is. All right, so the filtrate comes back up the ascending limb, and once we get out of the loop of Henle, the filtrate then moves into another convoluted portion of the nephron known as the distal convoluted tubule. And again, the distal convoluted tubule receives filtrate from the ascending limb of the loop of Henle. It is very reminiscent of the proximal convoluted tubule, though it's not as long and not quite as coiled as the proximal convoluted tubule. Like the proximal convoluted tubule, it consists of cuboidal, simple cuboidal epithelium, but the noticeable difference that you can make out even under the microscope is that the distal convoluted tubule does not possess microvilli, and again, you can't see individual microvilli with a light microscope, but you can see sort of a fuzzy border that they create. And so you look for that difference under the scope when trying to distinguish distal from proximal convoluted tubule. Here we see the distal convoluted tubule is the end of the nephron. Well, it's the end of a single nephron. But the tubular portion of the nephron, remember, is going to continue as a distal convoluted tubule, but it is as a collecting duct. So it is technically correct to say the distal convoluted tubule is the end of a single nephron, but it's important to realize it's not the end of the tubular portion of nephrons. They again converge to form the shared collecting ducts. And so the collecting ducts, again, are going to run essentially vertically between cortex and medulla. So fluid is picked up from many distal convoluted tubules, and the collecting ducts are then going to carry that filtrate towards the medulla, down into the medullary pyramid, and towards the apex of the medullary pyramids, terminating as those little openings called papillae. A papillary duct is a duct-like structure at the very tip or apex of the medullary pyramid, and it's formed, as you can see here, by several collecting ducts fusing together to form a larger papillary duct. You have about 30 of these papillary ducts that end in the tip of the pyramid as little openings or pores that again are going to leak out urine. So once the filtrate has flowed all the way through the collecting ducts, there's a little more processing that takes place on the way, but then when it finally leaks out of the end of a papillary duct in the tip of a papilla, at that point it is final urine, which then needs to be, of course, excreted from the body. The collecting ducts and papillary ducts, again, are lined with simple cuboidal epithelium without microvilli. So just to recap, the flow of fluid that's formed, it begins as filtrate at the glomerulus, moves to the proximal tubule, into the loop of Henle, into the distal convoluted tubule, into the collecting duct. The collecting ducts will terminate as several collecting ducts fuse and form a papillary duct. And you have about 30 papillary ducts that open as pores in the tips or apices of the medullary pyramids. The urine drips out and is collected by minor calyces. At this point, it's now called urine. Up to this point, it was referred to as filtrate. But once it leaks out into the minor calyx, it is now urine. And from this point on, we just have to get it out of the body. And so it moves from minor calyx to major calyx. Major calyces again fuse to form a renal pelvis. The renal pelvis then funnels down to become the ureter. Ureter carries the urine to the urinary bladder for storage. And when the bladder fills to a certain threshold level of stretch, that creates a reflex, the micturition reflex or urination reflex. And we then eliminate the urine from the body through the tube known as the urethra. So that's a very nice sort of summary of the flow of fluid from its origin in the glomerulus to its elimination through the urethra. And here we see again a picture of all of this. This is the tubular nephron. And again, just to review, filtrate is formed in the glomerulus. It is collected in the glomerular capsule or Bowman's capsule. The filtrate moves into a proximal convoluted tubule. From there to a nephron loop or loop of Henle. And notice the loop of Henle has thick and thin segments, the thick segment forming the beginning of the descent or the top of the descending loop and almost all of the ascending portion or limb. The thin segment forms the lower part of the descending limb and the U-turn that takes place. From the nephron loop or loop of Henle, we then move into a shorter distal convoluted tubule. When we get to this point, we're technically at the end of a single nephron, but multiple distal convoluted tubules are going to drain their filtrate into the collecting duct, which carries the filtrate down ultimately to something called a papillary duct, which represents the fusion of several collecting ducts. Just to point out where this is happening within a lobule, of course, as I said earlier, most of the nephron is up in the cortex area. The loop of Henle is the only part that dives down into the medulla. We get here, and that's the end of a single nephron. Multiple collecting ducts drain into this, multiple distal convoluted tubules, drain into this collecting duct. And note again, the orientation of collecting duct is to carry the filtrate down, down, down into the pyramid towards the tip, where several collecting ducts will fuse together to form a papillary duct. And again, you've got about 20 or 30 of these papillary ducts, which will leak the urine out into a minor calyx that surrounds and encloses the tip. Okay. Here we see a microscopic view of what this all looks like, and the glomerulus is very clearly seen as a spherical structure, and it's usually surrounded by a space, the capsular space, between the podocytes lining the glomerulus itself and the simple squamous epithelium. And here we see a squamous cell of the parietal layer of the capsule. Everything all around the glomerulus are tubules, renal tubules, and it's a combination of proximal convoluted tubules and distal convoluted tubules. Combination of proximal convoluted tubules and distal convoluted tubules. And the highlights are showing you the difference between proximal convoluted tubules and distal convoluted tubules, though I don't know that the magnification is great enough for us to see it in this case. The staining shows the distal tubules as a bit lighter in color than the proximal tubules, which are a bit darker in color, but that staining is not going to be necessarily a consistent feature. That is really dependent on the preparation. So what we want to look at is the inner surface, the luminal surface of the cuboidal cells, but honestly this magnification isn't great enough to notice that these will have microvilli, a more irregular shaped luminal surface, whereas these will have a smoother appearance. Again, a bit higher magnification where we see the glomerulus covered by podocytes, the capsular space, and then very nicely we see here the simple squamous epithelium of the parietal layer of the glomerular capsule. We see a proximal convoluted tubule here, and we see a distal convoluted tubule here. But again, not sure if it's terribly clear that we've got microvilli on this one and a smoother surface here. I don't think that's terribly obvious on this particular view. Here we see versions of the loops of Henle diving down into the medulla. So this is not taken from the cortex like the previous slides, where this is the medulla. Much of what you see here are thin-walled capillaries, the vasa recta. The thicker tubes here represent the thin segments of the nephron loop. The thinner walls here are the thin segments of the nephron loop, and then the thick segments of the nephron loop are these more cuboidal tubes. So we've got very, very thin-walled blood vessels, and then we've got thin segment of the nephron loop, and it's actually very difficult really to tell the difference between a capillary and the thin segment of the loop, because they're both essentially tubes of simple squamous epithelium. But we do see the thick segments, darker purple here, the thick segments of the loops of Henle. And then these larger tubes, remember, are going to be the collecting ducts, which also pass through the medulla. So the medulla is essentially going to be blood vessels, vasa recta, capillaries, thin segment tubes, thick segment tubes, and collecting ducts. Here we see a longitudinal view of the medulla, once again showing blood vessels, collecting duct, and different segments of the loop of Henle, thin segments, once again, the blood vessels and the thin segment are almost indistinguishable one from another. All right, there are two different types of nephrons in the kidneys. These are referred to as juxtamedullary and cortical nephrons, and this seems to be a good place to stop for today. We're just at about an hour and 5 minutes, a good stopping point. And so next time, when we get back together, we will describe the difference between juxtamedullary and cortical nephrons, and then finally move on to section 3 and begin talking about the physiology of urine formation. And we will start to talk in more detail about filtration, secretion, and reabsorption. So we're stopping today at objective 37, and we will pick up next time with objective 38. Until then, stay safe.